I sent you an email, Professor, and I was just waiting for you to reply, but I sent it to you like within like a minute or two of the meeting being disconnected.
And how long ago was that?
About 10 minutes.
Yeah,
So,
10
10
between
minutes.
5.
Had I gotten to the end of life elsewhere?
No.
No, we were still on the planetarium one about the Earth and showing the liquid water requirements it needed to sustain life.
Okay.
peptides and stuff. That's what we were on.
All right. Can you see that? Among
Yes.
multiple billions of
Yes.
stars.
Yes.
But I talked
Yeah.
about the Kepler Space Telescope.
Yes.
Okay. And what about the requirement for liquid water?
Which slide?
You were on the history of life on Earth, talking about how complex life only lasts, or was
Okay.
only in the last few 100,000 years.
Okay, so this is where it quit. Life on other planets and comparing it to how life likely started on Earth. Okay, well, the point of this slide is that on Earth, I'll just go to the next slide. On Earth, which formed about 4.5 billion years ago, and life first started about 3.5 billion years ago. And for billions of years, it was very primitive. No indication of any intelligence. And it's only been very recently that we've had complex forms. And here we have the primates 7 million years ago, which is a tiny amount of the time that the planet has existed. And humans, these are not humans, these are just apes. Humans have only existed on this planet the last 400 million years, which, put an arrow here, it would be right on 0.
I think we're looking at different slideshows, or different slides at least. I'm on one that says example of, I don't know how to pronounce that. I'm seeing slide 9 right now.
This is slide 9, and you were on slide 8.
It's showing slide 9.
It's showing slide 9.
Yes.
Sir, I think so. We are on slide 4 or 5 when you're there.
Here.
Just go back.
Okay, here's slide 4.
I think the slideshow that's being presented is the wrong one, because the screen's not changing. I can see your mouse moving, but not the slides.
Yeah, I can only see slide 9 is stuck on there.
All right. I'm going to stop sharing this screen and start over. Now what do you see?
Now we see slide 4.
Slide 4?
Yes, I think we're 3 down on the slide 7.
Yeah, we're down on slide 7 on this slideshow.
Yeah, that's where most of us got kicked off.
That's right.
Okay.
Actually, yeah, we had.
So now you see Kepler 186.
Yep.
Yes.
I think we actually finished slide 8.
Yeah, we did finish slide 8.
Finish slide 8. Okay. Then this is the salient point, that life on Earth has existed for a long, long time. 3.5, at least 3.5 billion years. But for most of that time, it's been unintelligent. So if we extrapolate that to life on other planets, and if we say the same thing is happening on other planets, then the chance of life, go back here, on Earth, only a tiny 1% of 1% of the history of the life has had intelligent life. So if that's happening on other planets, if life at this point in time exists, it's likely to be primitive, and it's not looking for us. We'd have to go looking for it. And this simply makes the point that the main problem in finding life elsewhere is distance. Within our own solar system, distances are fairly short in terms of light seconds and light minutes. But to the nearest star is 4.3 light years. Now that's a very long distance. And again, across our own Milky Way, 52,000 light years, which would take millions of years just to get across it at the speeds we can currently imagine. And Kepler-186, this most Earth-like planet known to date, is 500 light years from Earth, which is 2.5 times 10 to the 15th miles. So it would take approximately 10 million years, 10 million years to get to Kepler-186 from here. So that's not really doable.
Speed of light is 299,792 kilometers per second.
What's that?
Per second.
What, for a second, say it again. The microphone is fuzzy.
Never mind, it's okay.
All right, so the conclusions that I come to, and these are just my own opinions, I'm certainly no exobiologist, but the question, is there life elsewhere beyond Earth? The probability of it is very high. Why? Because of the Goldilocks phenomenon. The number of Goldilocks planets in the universe is probably very high, and that's just based on numbers. That's because there are so many different galaxies and solar systems out there. But the likelihood of detection by humans is very low. Now, why is that? Well, it's because the interstellar distances are so great, and we probably would have to go looking for life, because life elsewhere is not likely to be looking for us. And that's because life forms on other planets are likely to be very primitive, unintelligent at any given point in time. So given all that, the question is, will we detect extraterrestrial life before our own extinction? It's a good question. We don't know, but it seems to me the chances of it are not great, given what we know about life on Earth and when life on Earth first became intelligent. If the same sequence of events is happening elsewhere, then the chances of intelligent life being found by us or finding us are really quite remote.
But we don't know when our own extinction is either.
No, it's true. We don't know. We don't know when our own species will be extinct. Is that what you mean?
Yes.
It's true. Some days I think it won't be long. Other times I can imagine. Well, the other thing that our species will have to do in order to avoid extinction is to escape this planet before it becomes uninhabitable and establish life establish our species somewhere else. So we need really big space stations. All right. Any more questions about this? Comments? I hope it hasn't been too confusing. I'm going to stop sharing this and go to a different screen. Can you see
Yes.
classification?
Yes.
Organisms? Everybody see that?
Yes.
Okay. And so you've heard none of this.
No.
All right. All right. We'll start over. Let's see. We have about a half hour, so I might be able to get mostly through this set of slides. So how many different types of organism are out there? Over 1.8 million species have been identified so far, and that's not nearly all of them. And how do we keep track of all these different organisms? Well, we do it by giving them names, unambiguous names. And we classify them into groups that show relatedness. And relatedness is a key term here because by what criteria do we determine that something is related to something else? Remember the name Linnaeus. He is the father of the binomial method of classification. Binomial means two names, and the two names are genus and species. This is the formal scientific name. It's the name that everyone on the planet Earth uses to identify a particular organism. So, what's my binomial? Humans, Homo sapiens. This is Christina Nettie Parker, daughter of Catherine Nettie, who's the head of the biology department at Cuyamaca College. Binomial for cat, Felis catus. A killer whale, Orcinus orca. And this is how you use a binomial if you're writing. You use the two words, in this case the bacterium Escherichia coli, spelled out and italicized. Either italicized, or if you have no italics, you underline Escherichia coli with no underline in the gap between the words. And when you're using this in a paper, you use the full name the first time it's used, and after that you use the abbreviation, in this case, E. coli. And very often in popular media, when you see this bacterium referred to, it's just called E. coli, because that's easy. Here's an example of use. Streptococcus pyogenes is the bacterium that is known to cause strep throat. The antibiotic amoxicillin can be used to kill most strains of S. pyogenes. But you only use the S for the genus name the second time it's used and subsequent to that. Humans are natural classifiers. And you think of how you classify things all the time. For example, look around your room. How do you classify your furniture? Probably you've got bedroom furniture and you've got living room furniture and kitchen appliances. They're all classified into groups. We do that just to make, so we can continue to make sense of our environment. Now, Aristotle, for example, the Greek philosopher, classified organisms as either plant or animal. He said everything on the planet is one or the other. There are a lot of organisms that don't fit into that system. And so over the last 300 years, we've developed other ways to classify. Currently, all life is classified into one of three domains: bacteria, archaea, and eukarya. Now you can see the branches here: the domain bacteria, the domain archaea. Domain eukarya is divided into plants, animals, fungi, and protists. We'll come back to protists a little later, but they are, it's a grab bag of anything that isn't anything else. It's called a protist. So classification involves putting things in boxes and then dividing those boxes into smaller and smaller boxes. So here you have three boxes representing the three domains of life. If you take the kingdom, the domain eukarya, and divide it into kingdoms, you have the protists, the plants, the fungi, and the animals. Four boxes from one. And then if we go divide that further, you have everything that you see here, the animal kingdom. And it comes down to this. And this is something that you should regrettably have to memorize, but you really should know this. A domain, kingdom, phylum, class, order, family, genus, and species. Different people have different ways of trying to remember that, but it is important. So let's look at an example of classification.
Professor, just really quick on slide 15, which part do we have to memorize, the whole slide, or?
Yeah, know this classification system, because this is standard for all forms of life. The domain, the kingdom, phylum, class, order, family, genus, and species.
I appreciate it. Thank you.
We're going to take an example looking at three species, 1, 2, and 3. They're all in the domain eukarya. They're all in the kingdom Animalia. They're all animals. They're all in the phylum Arthropoda. But when we get down to class, they diverge. So species 1 is a crustacean. The other two are insects. At the level of order, again, species 1 is Decapoda. The other two are Diptera. Then at the level of family, they are distinguished from each other. And at the level of genus and species, they become recognizable to us. So Homerus americanus, Drosophila melanogaster, and Aedes aegypti, what are they really? I wonder if this is why it keeps, do you see this? It says upgrade, I'm running out of time. Meeting will end in 10 minutes. I've never had this before.
It doesn't show on my screen.
What's that?
Yeah, it says it will end in 9 minutes.
Okay, well. Huh. If it ends in 9 minutes, so be it. We'll pick it up next time and I'll try to figure this out.
I think Zoom only runs in 40-minute sessions. So to go past a 40-minute, you have to create an account. And I think even buy, like a Zoom account. Once you pay for it, you can run as long as you want.
Okay, well, it's weird that, I did this all of spring semester and the first 2 sessions this semester without the problem. So I must have my settings wrong. It's causing this problem. Anyway, I'll try to figure this out. I don't want to. I don't want to go into it now. And if we get cut off again, we'll just pick it up next time. Well, on second thought, if I can restart the meeting, if it cuts off and I can restart it like I just did, I will do that. Okay, classification of the 3 species. Remember, we got down to genus and species. They're all different. Well, the first one is a crab, Homarus americanus. Second is Drosophila melanogaster, the fruit fly, which is a genetic model for studying genes in organism. You probably know more about genetics because of Drosophila than any other organism. And Aedes aegypti is a mosquito. Now, what criteria are used to classify organisms? It's based on phylogeny, which are evolutionary relationships. But the most important criterion for classifying organisms now is the ribosomal RNA sequences. And let me just say a little bit more about that. So the contemporary bases for classification of organisms are, first of all, classical morphology. What do they look like? What is the size? What is the shape? What is the color? And you can look for relatedness among groups by those criteria. But much more precise is the sequence of ribosomal RNA. Why that? Why is that so useful for classification? Well, first of all, it's found in all organisms. There's no living thing that does not have ribosomal RNA, from bacteria to blue whales to sequoias. The other important thing is that some regions in the sequence are highly conserved, and they can be used as reference points in biometrical analysis. The other important thing is that some regions are highly variable. And these highly variable regions can discriminate among species. So if you look at this structure of ribosomal RNA molecule, and the colors, blue means not conserved at all. The red means highly conserved. So if you look at just these 2 amino acids here, that means they're highly conserved. That means they're found in everything from bacteria to mice to oak trees. Every organism has the same 2 amino acids at that position. Whereas if you look at the blue regions, for example, this spatial region here, it's blue, which means that it's not conserved at all, and that is a region which will tell you the difference between one species and another. And it's whenever you see a phylogenetic tree like this, it's almost always based on ribosomal RNA sequences. So here you see the 3 domains, the bacteria, the archaea, and the eukarya. Most of these names you will not recognize, and you don't have to remember them. Just remember the domains. Remember that the archaea, the ones that we know most about, are extremophiles. These are produce methane. These grow in salty conditions. These are very heat tolerant. But I'll say a bit more about archaea in a bit. Among the eukarya, the ones who we're most concerned about are the plants and animals, and fungi to some extent. Fungi, it turns out, are more closely related to animals than they are to plants. The domain bacteria, they're the simplest organisms, they're prokaryotic. And this shows you the 3 basic morphologies that bacterial cells have. They can either be spirals or rods or spheres.
Question, what does phylogenetic mean?
Phylogenetic, I To be precise, I should look up the precise definition of it, but it means relatedness among organisms. So go back here, it's a phylogenetic tree. It's the relatedness of bacteria to each other, the relatedness of bacteria to archaea and eukarya. So the groups, this is a phylogeny. Eukarya has a phylogeny within its tree. I would encourage you to look up the precise definition of phylogenetic, which I will do myself after the class. Okay. Well, this is all you need to know about the domain archaea. We're not going to discuss them anymore. They are prokaryotic. They are single-celled. Many are extremophiles, as I just said. They grow in salt lakes or thermal vents and other extreme environments. Those are the ones that we know about, but we now know that they're really ubiquitous. They're everywhere on the planet. And the reason we don't know much about them is that most of them are not culturable. And we know their existence because of metagenomics. Metagenomics is the sampling of raw materials, for example, soil or a cow's gut or seawater. You extract all of the DNA from such a sample and sequence it. And from the sequence, you can identify organisms. And so most of the archaea we know from metagenomic analysis. But we know that they are abundant in animals, especially in the gut, the mouth, and the skin, including us, humans. We have archaea all over ourselves. None of them is known to be a pathogen of anything. Most of them host viruses, and they're sort of an intermediate, genetically and metabolically. They're intermediate between bacteria and eukarya. Here's an example of an archaeal virus. Here, different types. Thermophiles grow in hot springs, acidophiles in very acidic environments. Halophiles grow in salt lakes. And these are some of the shapes that archaeal cells can have. Here's scanning electron micrograph of one type. Here's another type.
Professor,
Yes.
quick question on the quiz. So is the quiz still going to be due tomorrow since we had the technical issue problem? We didn't get to finish the presentation today? Or not tomorrow, Wednesday. Sorry about that.
The quiz. Okay. Yeah, the quiz will be Wednesday. I haven't made it up yet. So I will make sure that the questions are based on what you actually hear today and what you've heard in previous lectures. Okay, the domain eukarya. These are cells with nuclei. So they're animal
We
cells.
have less than 1 minute for the screen to show.
What's that?
It says we have less than 1 minute for the screen to show.
Oh. All right. Then I'm going